Hi, everybody. Hope you're enjoying your snow day. Just want to take a minute to go through and talk to you about the bioinformatics lab. Um, we collected these samples, we processed these samples many, many weeks ago, and this week is an opportunity for you to finally get the answer as far as what your species is. So we've already collected our organism, we extracted the DNA, ran PCR on it to give you a little bit of a review. Remember, PCR is basically like photocopying DNA. We made many, many copies of it. We then ran electrophoresis to make sure that there was some amplification, and I remember just about everybody had some sort of amplification, so we did real well there. So we sent those samples off to a company, and they sequenced it, and now we're going to look at the bioinformatics end of things. Meaning that how do we take that sequence and turn it into a species, right? So how do we figure out what the species is that we were looking at? All right, so we collected the specimen, extracted DNA, amplified, checked it out. We sent it off for sequencing. We're going to talk a little bit about how that works today. Then we're going to go through data analysis so we can take that sequence, trim it, line it up, do the things we need to, and then we can send it through a barcode library that tells us what our species is. There's other information in that sequence that'll also allow us to set up a phylogenetic tree and be able to look at how does our organism relate to the other organisms that we have collected. All right. So how do we determine that DNA sequence? So remember, DNA contains a unique code that can identify species, right? That code is made up of 4 letters, right? Or G, T, C, and A. The order of those letters is what is going to determine our sequence, right? So we can't see these letters with our naked eye. Even with a very good electron microscope, we would not be able to read those letters. So we've had to come up with clever ways to do that. So they come up with a clever way to do this, to figure out what order the bases are in. It's this thing called Sanger sequencing. So what they do is they do PCR, right? So they make copies of it, but they put these special bases in. Now these bases, right, each A, C, T, and G each have a particular fluorescent molecule attached to it. So in this example, the A's would have green fluorescent protein, C's would have red fluorescent protein, T's would have blue fluorescent protein, and G's would have orange fluorescent protein, but they depict it as black because it shows up better on, you'll see, our readouts look better if we throw orange in there because sometimes we can get confused with the red. All right, so what they do is they run PCR, and there's a mix of both regular bases and these special fluorescent bases. So the polymerase will go along the sequence and make copies until it accidentally grabs one of these fluorescent bases and puts it on there. Because the fluorescent bases are different, the polymerase will pop off of that sequence, right? It'll fall off right there, right? So for example, this one, it hit the first G in the sequence, grabbed one of those fluorescent bases, so boom, all it put on there was the G, and then it fell off. Next one, the G is a normal base, and then it grabbed a fluorescent base for the T, boom, fell off, right? So it goes through, it does this all the way down, hundreds and hundreds, if not thousands of bases, right? It does this, it keeps making these randomly, and so each, it'll keep making longer ones, shorter ones, right? So eventually, law of averages, randomly, we've got the entire sequence has been copied, right, all in different lengths. We then send that through a machine, all right, so it's this thin tube of acrylamide, and so this acts like the gels that we ran, and so our sequences are going to go through, and if you remember in electrophoresis, our shorter sequences are going to move faster, so the first one through is going to be the smallest sequence, and so the first one through is going to be that G. We've got a laser that makes the molecule fluoresce, makes it shine. The detector picks it up, all right, and that reads what base is going through. Now the next biggest one's going to go through, and so what it's telling us is like, okay, the first one is G, next biggest one's going to go through blue light, it's T, next biggest one's going to go through red light, it's C, next one's going to go through green light, it's A, right? So if we're going to do this for the entire sequence. So the sequencer reads those fluorescent tags. It gives us peaks, all right, in the reading to show us which ones are there. We run this through an algorithm that goes through and determines what the code actually is. And so now we've got our code. And since each of our species have a unique code, we can compare those sequences with each other to see what identify our invertebrate. All right, so what it's going to do is go through and compare itself with the ones that are already in the database. So we've got our code that we determined, GTCA. If we look on our example, that is the mayfly. So it allows us to go through and use that DNA barcoding, figure out what our species is by comparing it to thousands and thousands of other species that folks have put into this database. We can also determine phylogeny. So phylogeny is evolutionary history. The way we think of it is a family tree. All right. So how people often build those, looking who are your parents, who are your grandparents, who are your great grandparents, that sort of thing. It's the kind of same idea except for different species. So we can use this code to compare relationships. The reason we can do that is the more closely related an organism is to another organism, the more of their sequence that they're going to have in common with each other, right? So we can compare sequences, see how closely they match up to each other, and say, okay, that gives us an idea of how closely related those two species are, right? So we start with our mayfly, we can compare our differences, and we can come up with assumptions about how related each of those are and what their evolutionary path is, right? So we've got this common ancestor bug. We can see that our dragonfly is most closely related and that our mayfly, our maggot, and our caddisflies are all equally distantly related from that common ancestor. And we can get an idea of what their evolutionary relationships are. All right, so what we're asking you to do is go ahead and use the protocol of the lab. All right, I'm going to upload the data that you need onto Brightspace and have directions there for it. I'm also going to set up another video that walks you through how to go through and look up your sequence online. So just take your time and walk through it. It's relatively straightforward, and you should be able to get some good data as far as what your critter is that we collected all those weeks ago. So first off, I'd like to thank you all for a wonderful semester. Had a great time teaching you, and look forward to seeing some of you next semester. There is no post-lab quiz this week, so don't worry about that. Please go and fill out the course evals. All right. It is great feedback for me, gives me an idea of where I can improve, what you guys like, what you didn't like, all that sort of thing. So I look forward to seeing a bunch of feedback from all of you. Good luck with the semester, and yeah, we'll see you all later.